Welcome, everybody. My name is Tommy Soros. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And today on Pathfinder, I have the honor of welcoming Dr. Bruce Cooper, who's the director of the Metabolite Profiling Facility here at the University. And has been such a great journey so far in speaking to all these scientists so far on Pathfinder, Bruce. But I got to tell you, I've really wanted to speak to you because you have a very unique story. I've known you for so many years, and I know your story that I really want everybody to hear here. The uniqueness is that most people, they go from academia and they move to industry, never to come back to academia. And your story is different because you actually came back and you've been working as the director of the Purdue Metabolite Profiling Facility for so many years now. And I want everybody to hear your story and just to hear the trajectory of your career so far. So thank you for doing this, Bruce. It's so nice to see you and talk to you again.
Oh, I'm happy to do it, Tommy. Yeah, you're right. I was in industry for 10 years. I worked for Eli Lilly and Company, the big pharmaceutical company, right after getting my PhD. So I didn't do a postdoc. I knew when I went into graduate school that I wanted to work for a pharmaceutical company. That seemed like a nice, noble thing to do, right? Make medicines, help sick people. However, I knew I didn't want to work where it was really populated and crowded. I knew New England, or like Boston or something like that, would never work for me. I just don't like that. So I said, okay, I want to work for a pharmaceutical company, but I really don't see myself working on the East Coast or the Pacific Coast. Well, that knocks out the majority of the job opportunities. When I got my PhD, I was 28 yeyears old, and I had only lived in a state that bordered the Atlantic Ocean. So all I kind of knew was East Coast, and that was kind of one reason I knew I didn't want to have spend a lot of time there as far as raising a family and stuff. So then I looked, and there's only a few companies that really are more centrally located. Eli Lilly here in Indiana being one of them. So that's what brought me to the state, and that had to be 30 years ago, I guess.
Wow, that's awesome. That's awesome.
So I came out here. They talk about all the stresses, some of the most stressful things that can happen to you. And within about a 2-week period, I had a lot of those because I had to defend my PhD. I got married 2 days later, and then I moved. I think that's on the list. And then I started a new job. So it's just like
Wow.
and that was January of 1993. Yeah, but that's what brought
My
me to
gosh.
Indiana was Eli Lilly. So I did work there for 10 years. I was in the drug development section. So really, when you think about a pharmaceutical company, the evolution, the pipeline is that you start with discovery, and that's a whole entity unto itself.
Sure.
And then when things show promise, they move into development, and that's where I was. So in development, we were trying to make the active pharmaceutical ingredient, but do it in such a way that the buzzword was manufacturable. And what that meant was I'm an analytical chemist, but I work side by side with the organic chemists who are thinking about the synthesis. And what I learned, it was interesting for me. If you're making something in a chemistry lab, like I was used to at the time, when you're making like 1 gram, you could use very high temperatures. You can use catalysts and solvents like chlorinated solvents, methylene chloride. And it works great when you're doing it on a 1 gram scale in the hood. But when you start thinking of being manufacturable, now you're talking, okay, I got to start with cheap starting materials. But things like catalysts and chlorinated solvents don't fly when you're making, 1,000 kilograms in a bag, which is what we did. And I say I'm picking on the chlorinated solvents because there's anything that's considered very volatile it was an issue just because of environmental concerns. The way it
Sure.
works on a large scale is they say, okay, you brought in so much, say, methylene chloride, um, when you started this process, and you were able to recover so much, anything that's missing was considered a loss, and it went into the environment, even though that was probably not really the case. And then EPA and others would penalize us for that. So yeah, we were had to make the organic chemists I work side by side with are trying to make things manufacturable. And because you're limited to the temperature extremes, the pH extremes when you're working on a very large scale in these manufacturing buildings. And so as a analytical chemist, every single step of the process, from your starting materials to your synthetic intermediates to the final, we call it the API, the active pharmaceutical ingredient, has to have an analytical method where you're looking at the purity, you're looking at the impurities, and then we typically looked at the solvents. That's all the steps. And on average, when you look at when you purchase a pharmaceutical, a small synthetic molecule, it probably has about 7 unique chemical steps. So I was working on that type of methodology. But yeah, as far as then what got me to Purdue, that was in Lafayette, Indiana. The Eli Lilly had a plant site located here. They have plant sites throughout the whole state of Indiana. But there was a lot of talk of closing that site. It was built in the 1950s, or I shouldn't say, well, there was talk of closing it, but then they ended up selling it. But they made it clear that they were going to move all the scientists from the Lafayette site down to Indianapolis, about an hour away. And my wife and I talked, and we said we like Lafayette, Indiana, and it just for a number of reasons it didn't really work for us to move, and so I resigned from that position. And about that time, they had just built this Binley Bioscience Center here on campus. And I could remember that I was interested in working here. And as part of the kind of the building, the initial construction, they also included the instrumentation. So there were 4 mass spectrometers in this metabolite profiling facility room, but they had no staff, no, really none. So they had bought, some faculty had recommended what instruments to buy, but they had nobody to use them. And so that was the carrot that was dangled in front of me at that point in time was, if you come here, you get 4 brand new mass spectrometers. And in my work with Eli Lilly, I was doing HPLC with UV detection. That was the standard. We had mass spectrometers. It was a group of just 3 individuals, but you never ran them yourself. You submitted the samples to them, and they'd give you back the paper with the results. And we did that to identify impurities. But I had never done it. But I thought that's pretty cool technology. I consider myself a chromatography person by training. Always wanted to learn mass spec. So, hey, here was an opportunity. So I was hired in, as you said, a long time ago. I was thinking about it. It was 17 years ago. I can't believe that.
Wow.
And I remember the announcements came out that Bruce Cooper, a mass spectrometry expert, has joined the Binley Bioscience Center. And I went to my director at the time and I said, I'm not a mass spectrometry expert. I've never done it. And he's like, oh, but we hired you because we think you could learn.
Yeah.
So I was going home and reading everything I could, and yeah, and I realized I really do enjoy it. So yes.
That's awesome. What a great story. What an awesome
Yeah.
story. Bruce, but what was your training in? What was your PhD in? And it was in North Carolina, is that right?
Yes, I got my PhD in North Carolina, University of North Carolina at Chapel Hill, working for Jim Jorgensen, who was a big separation scientist guy. He's now since retired. And so his niche was to take separation techniques and miniaturize them. He was the one who invented a separation technique called capillary electrophoresis, which the
Oh,
proteomics group has. They're commercially available now. All the devices were homemade when they were in the laboratory, but it's a separation technique. which is what most people in the lab worked on. I did not. I worked on miniaturizing HPLC chromatography. So we had everything was homemade. We would make our own columns. The separation column was very small. Now I'm using separation columns where the diameter is now 2.1 millimeter. At Eli Lilly, they were 4.6 millimeter columns. They were bigger. But when I was in grad school, the columns we were making were 50 microns in diameter. So
Oh, wow.
it was very, very miniature. And I was doing electrochemical detection because it's very sensitive. And you'd say, well, why would you want to do that? Why would you want to miniaturize that? The application was we were doing single cell analysis, which at the time was really cutting edge. It was really novel. When I started, we were the one person in the group before me started the single cell analysis, Robert Kennedy, who's the professor at the University of Michigan now. And we were starting with snail neurons because it ends up snails don't have brains. Technically, they have, I think it was, if I'm remembering, it was a ganglia, but they
Yeah.
have something called a giant dopamine neuron. And so we were getting snails, I remember illegally. There was some guy in California who was going out and capturing these snails and putting them in a box and mailing them to us in North Carolina, which I think from because of the damage they can do to plants, I don't think that was even legal. But I would get this box, open it up, cardboard, and there'd just be full of snails all over the place. And we had aquariums. But I was doing that work under a microscope. I learned how to dissect the snail and find this giant neuron, single neuron cell that was dopamine. And then we tease it out, and then we were of all homemade stuff. We made little tiny vials and devices where we could pull up that single cell in solution and put it in a vial, and then we mechanically lysed it and then inject it onto our chromatography system, and then we would see a peak, for dopamine. I mentioned we had an electrochemical detector. The neurotransmitters are electrochemically active, just naturally. We didn't have to derivatize them or anything. And if you have analytes that are electrochemically active, it's a great detector because it's super sensitive. But
Yeah.
then my project, somebody else, a professor from Indiana University, moved to Chapel Hill, and he brought with him cultured cells. And he was looking at adrenal cells. He was an electrochemist, so he was looking at epinephrine and norepinephrine. And so we decided to kind of abandon the snail stuff after my first year and switch. And then I was looking at, so much smaller. These neurons, I think, were 120 microns in diameter. But then we went to culturing adrenal cells in grad school. They were just 10 microns in diameter. So we were doing that. Overall, it was a lot easier to work with cultured cells, but everything else was pretty similar as far as teasing them out. And then we were looking at those neurotransmitters and quantitating that at the single cell level.
And do you remember working with him? What any words of wisdom that he had as you were thinking about what am I going to do next? Like, where am I going to go? And you thinking, okay, well, is the coast the destination for me?
Right.
Stay here, or you obviously looked into the Midwest and in Indiana to Eli Lilly as a place where you could like blossom, right? And like continue to work on your career. What did he say anything to you like, hey, before you go? Or like, I mean, because as the inventor of capillary electrophoresis, this guy must have been like, man, like awesome, eh?
Yeah,
To
it was my graduate student career was great. I look back on it as probably where I peaked, just because of the people I worked with were real smart, very motivated, very independent, and we were just doing some things that were really novel. But my professor, Professor Jorgensen, yeah, what was I remember was just when you got into a brainstorming session with him, it was really great. He was very mathematically oriented. He knew all the theory for doing chromatographic separations really well.
yeah,
And so we would often go to the blackboard and just start doing the math. Well, what happens if we do this and blah 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 blah. So that was fun for me. Just that when he postulated an idea, it always went back to let's think about it with all the equations that are involved with operations and see if this makes sense.
Yeah. Yeah.
And so that's for capillary electrophoresis. That lab at the time I was there, something that's very common now is UPLC, or ultra high pressure HPLC, and that came out of his lab right about the time I was leaving. There was one new student coming in, and he was assigned to, hey, can we build one of these systems, where at that time instruments would probably run at about 30 bar, excuse me, maybe like 300 bar of pressure, maybe not even that high. But can we take it up maybe 10 times higher? And again, the mathematical equations said that you would get much better separation efficiency if you could go to smaller diameter particles, because HPLC columns are packed with a resin, and the resin has a certain diameter. At the time, it was 5 microns was standard. But if we could use smaller packing, the theory said that you could get much sharper peaks. So if you have a very sharp peak, so it's very narrow, skinny peak, then it's very tall, you can pack a lot more of them into a certain amount of space. So for complicated biological samples, that was the way to go. The problem was, if you have a small diameter resin, the pressure required to push liquid through that resin goes sky high, and there just weren't any instruments to do that. So we were working on building it. And in grad school, we all had our jobs and our responsibilities, and one of them was I was like the fittings guy. So when we had tubing and connectors for the tubing and stuff like that, I always made sure we had the right stuff. Well, now it was getting into, can we build a system that can go to very high pressures, and will the tubing and the hardware withstand it all? And so I kind of did the research and had some of the documentation of what the manufacturers were selling. It says, actually, this stuff that we're using now was rated for super high pressures that nobody ever goes to. So yeah, this stuff will all work.
Yeah.
At the time, we were concerned about it. So when we built the first very high pressure system, we also built a containment wall, a box around it, using like 1/2-inch steel, because we were talking and we said, I think my professor on TV, I saw this thing once where they were cutting metal with a stream of high pressure liquid. I think it was just water, but it was cutting metal. And he goes, I don't want to kill anybody by making one of these systems. So we built this box around it, fearful that what if this thing explodes, or if you get a leak, is the stream going to shoot out and cut somebody in half?
Yes.
But then we kind of realized, well, it's not going to explode. You think of compressing a gas at a very high pressure. If you get a leak, things can literally explode. But with liquid's not very compressible. So if you get a leak, it just kind of forms, a little drop for the most part.
Right.
And all that stuff we did safety wise was not necessary, but we didn't realize it at the time. But a lot of good memories of doing that.
Oh, that's great. So it sounded like it was really hands-on experience, and hands-on not only from the mechanical point of view and building these HPLCs and building the separations units, right? But it also seems like he had a lot of good theory behind the work, and was teaching you really how to be elemental about it, and scientific about it, right? And it seems like that must have prepared you well for your next step, which was it directly to work at Eli Lilly when you moved?
Yes, I
And
didn't do a postdoc, because at the time I was thinking, postdocs is if we want to go into academia, and then I didn't want to do that. I think I was what I was most concerned about was all that the grant writing and the uncertainty about the future. That kind
Yes.
of scared me when I was a graduate student. But well, if you go into industry, you can get a job, and then you don't have to worry about the money that much,
Yep.
and you have stability. So that was my thought process at the time, and it probably wasn't all that incorrect. So yeah, going into industry, you mentioned I've seen both sides. So 10
Yeah.
years in industry, and then almost 20 years here at Purdue. And so when I meet with graduate students and they're thinking, oh, gee, what am I going to do next? I do think I'm in a good position to say, well, if you go industry, here's what you can experience. And if you're going to go academia, here's what you can
Yeah.
experience. And it is absolutely different. When I first started, I'll tell you one story. When I first started, in the group I was in at Eli Lilly, they said, we want to have a preparative HPLC system. So the advantage of that is you can load a very large, it's the exact opposite of what I was doing in graduate school with a single cell. Now we want to load a large amount of material on. And the purpose of that is then you can collect some of the smaller peaks that you see in your separation, or the lower abundant compounds, and you could isolate them and then go to some other analytical techniques to determine what they are, because identifying impurities was certainly important. Because if I can go back to the organic chemist and say, in your process, yeah, you're 95% pure, but these impurities you're making are this, this, and this. They
Sure.
they think, oh, okay, I'm clearly over reducing my starting material. So it gives them some direction how to improve the chemistry or improve the synthetic process. So
Yeah.
we needed to build something. And they said to me, we had one of these instruments a real long time ago, but I think some parts were broken and some parts were just put up on a shelf. Can you bring it all back together? Well, I found a part that was part of a larger HPLC or preparative HPLC system, but it was broken. And so when my natural thought was, okay, you know what? I could go to the hardware store and get some things to fix this. Because that was my mentality. In grad school, everything we did was built, and my professor loved it if we were doing stuff, buying parts from hobby shops and hardware stores and things like that to construct this stuff. And I had that mindset, and so I was taught I was probably only there 2 months, and I had a meeting with my boss, and I said, well, this part was broken, but I went to the hardware store and got some washers and was able to do this, and he looked at me and he goes, why on earth are you wasting your time doing that? He goes, if the thing's broken, buy a new one. And I'm like, what? He's like, just spend the money and buy a new one. And that was a paradigm shift for me. I use that as an example of an industry. Money really is secondary. The drive is to get the job done and get it done fast. And that was a learning point for me, because at the time I was there, if a drug was super successful in the market and given the identifier as a blockbuster, at the time it meant it was over $1 billion in sales. And they said, if you can get your drug to market one day faster, um, and it's $1 billion a year in sales, do the math. Think about how much money the company's making in a single day. If you have to
Yep.
spend $2,000 on something, that's kind of trivial. And so
Yep.
there it was, money was buy what you need, but get the drug to market as fast as possible. Where in academia, you're far more cost conscious, and you're not as much on a timeline. I'm sure you want to get a paper published quickly, or I got a conference coming up and I want to have some data. Yes, we have timelines, but it's not the same. In academia, you're far more working with a limited budget, and graduate students are, you're here for 5 years, you got time to do this, and so the urgency is not there relative to industry. So that's the difference. One of the
Yeah.
differences, I think.
And was it tough to apply for that job from North Carolina? In terms of like, do you remember at all the interview process and what you had to do? And
I remember at the time the way it was, and I have no idea how it is now, but companies would come to campus and you'd interview on campus, and they'd interview a number of people. So you'd hear a certain, could be Merck or Eli Lilly, somebody is coming, and you'd sign up to speak with that person and submit your resume. And then those industry representatives would probably be there for about 2 days, and you'd have certainly no more than a 30-minute, maybe it was a 20-minute time slot, 20 to 30 minutes, um, where you kind of pitch yourself and talk about your research. And then you would hope you would get, the next step would be to get an on-site interview. So then you could go out and see the facility and see the town you'd be living in. And so that was the process. Of course, this was all before anything electronic and internet the way we know it today.
Sure, sure.
Uh, but yeah, I think it was a good match. I mean, when the work at Eli Lilly was HPLC with UV detection, and so I came with a big separations background, so that was a good
And
fit.
so were you able to kind of hit the road running? I mean, this epiphany or this kind of conversation that you had with your boss, and they say, "Hey, what are you doing? Let's just go buy a new one,"
Yeah,
and explaining that. So that shift, I mean, did that give you wings to like, okay, now I can really do some good stuff here and get my preparative HPLC
yeah, yeah,
going and so
yeah.
on, right?
Yeah, yeah,
Tell
yeah.
me.
Yeah. I mean, we had to put together like 4 separate units. There's the auto sampler, there's the pump itself, and then there's an automated fraction collector. And I think they had maybe 2 of those pieces already present. Other than there's the column itself. I think they had 2. I had to buy one, but then the challenge was to get them all to talk to one another.
Mm,
Um, um,
yeah, and
and
synchronize, right?
Exactly.
Synchronize.
Yeah, because they
Because
all
you would
weren't from the
only
same
want it to collect.
manufacturer.
That's right.
Yes, yeah. It was all done through time.
Time, yeah.
Right. And so yeah. I think I definitely was able to hit the ground running relative to my peers, considering that lab was at Eli Lilly was considered a separations lab. Most of the PhD scientists did not have a separations background. We used to joke that most of them were electrochemists. So I think I was able to hit the ground running just with my experience. I was a better match for what everybody was doing there.
Yeah,
Yeah,
yeah.
yeah.
So tell us a little bit about that experience in with the team and the chemists and the analytical
Ye-
group. What was the dynamic like?
That was one of the enjoyable parts of the job, is you were definitely on a team, and that was new for me. In grad school, I was the only one doing my kind of projects. Some of the my friends, there might have been 2 or 3 of them. They're all kind of working together to get something to work. But for me, I know I was flying solo. And then when I got to Eli Lilly, yeah, I really had to. That was an adjustment for me. I had to work with the team mentality, because I mentioned already that I work closely with the organic chemists who were changing the synthesis. And then they'd give me samples that I tried something new. How well did it work, Bruce? And I'm the analytical person, and we work together very closely. But you were on a project, you were assigned to normally just one drug that was in the pipeline. But the team required a lot of different disciplines. Here on the plant site, when you did go to scale things up, and there was a pilot plant facility before you get into the larger actual manufacturing facility, you worked with chemical engineers, which I had no experience with. So you kind of learned how they think and what are they interested in. You worked with the toxicology people. You had to give some of these impurities I talk about. identifying the tox people, the rule was if anything was present at 0.1% relative to the drug you're interested in, 0.1% or greater, it had to go through tox separately. So that was one of the reasons we were trying to, going back to the preparative HPLC, we're trying to isolate these so they could go through tox studies. But then you learn how the tox people are thinking. And occasionally, not all the time, but you even start, even though the drug wasn't for sale, they get the marketing people involved, and you start realizing how big a sales force is this going to have. And so yeah, you're right. You definitely broaden your horizons when you start working with some of the other individuals. And then of course there's the whole the business side of things. You work with people who aren't scientists at all. They're the MBAs assigned to the project, who are thinking about your timeline and the costs of everything that's going into bringing this compound or drug to market. You work with the clinical trial medical doctors, the MDs, because at my, I was saying earlier how there's discovery and then there's development, which is where I was. So we were really supporting the clinical trials, make material that's going to go to clinical trials to see if it's eventually going to make it to market. And then if things were promising enough, you would write government documents for approval. That was, and again, this was before everything was submitted electronically. But when the document left the facility to go to the FDA in Washington, D.C., that says, here is everything you require of us, because we have to have permission to sell the drug. It literally went into a semi truck, an 18-wheeler semi. It was just paper. And I would write just a tiny fraction of the whole, but we would have to have all our methods, and we'd have to show how we validated them. If we developed a method, its linearity was adequate, the precision was adequate, accuracy was adequate, and stuff. So we had to do that with all our methods, but ours was just the tiny. It had all the clinical trial data was involved in, but that was just the volume of paper. And then once it was submitted, you sat on your hands for about a year. Because the FDA, you didn't give them stuff in pieces. They kind of got the whole bolus all at once, and then you waited for them to say yes or no. You waited with bated breath. And then the process was, but during that time, you'd be optimistic that you were going to get approval. But I was in the development group, so our methods got transferred to the next step in the pipeline, which was quality control. So they were the ones who handled the marketed items. So once something was being sold, it went to the quality control when they were making it routinely in bulk, and it was no longer kind of a development experimental project. It was, here's our process, here's the final. That was the quality controls group. So we were very involved with transferring the methods. We'd have to say, because there was a quality control group in the building next door if the drug was going to be made here locally. At the time, Lilly had a big manufacturing site in Puerto Rico, and it also had another one in Ireland. So you never knew where it was going to go. But once you found that out, then you had to transfer your methods, and that involved. We talked about different groups. That involves the statisticians, because we would have to take maybe like 5 lots of drug, and we'd analyze them in our lab and get results. But then you'd have transferred all your documents to another lab, and they would get the same samples and run them. And then the statisticians get involved and say, are these results comparable, or is there a bias? Because one thing we were responsible for putting together was the specifications. So we'd say for the final active drug ingredient, when it's being manufactured for the next however long you have the patent, maybe 15 years, the purity has to be greater than, and it was usually pretty high, like 99.3%, and the impurities have to be lower than this, and things. So the statisticians would have to get involved to say, are these accurate numbers? And that was fun. That was fun to realize, just because all my life I've been working like with the biology people and the chemistry people, and now it opens up your horizons. To get into industry.
No, absolutely. And what's so fascinating, Bruce, is that you had your role, but in in that role, you got to see so many of the mechanics of how things move in that field. And you've just said a few of these tidbits that I'm hoping people are listening, because there are a few lights that have opened up in my mind as to, oh, that's how it really works. And who knows if things have changed. I know that the FDA process has changed from the devices side to be more, let's have a conversation and you tell us what you think you should be doing and we'll say yes or no, and
Mm,
then you can keep this relationship going until everybody's in agreement that what you're going to be submitting is good to go, type of thing. But
right, right.
because they don't want you to be sending reams of paper and not have all the qualifications that they want to see there. But so interesting to have it from your perspective, even as the analytical lab researcher or lab
Mhm.
technician that would provide this type of purview, but also being able to see where you saw that connection in the company. I'm assuming that pressure was high too, in terms of like deliverables and the timing. And as you
It
said, oh my
was.
gosh, we got to get the drugs out of here, and the sooner we can do it, the better it can be, and more lucrative, more profitable.
That's right.
um, and now the company shifts. Okay, everybody's moving down to Indianapolis. You don't want to go. It's not really good for you and your family. And you find this opportunity to now get into this brand new world of mass spectrometry
Right,
versus high performance liquid
right.
chromatography, high pressure liquid chromatography. Now, that must have been scary too. Like, oh my gosh, now I got to learn this whole new field. But it came along, if I recall at that time, it came along with your breadth of knowledge of separation science, which was an incredible gift to us at Purdue. And I remember working with you closely then
Mhm.
when we were trying to look at least from my part, the different metabolites that were being produced in plants at a time, but also in so many other different organisms. What was this opportunity that you said, well, like, I'll give it a go. I'll read as much as I can. It must have been so exciting to be able to delve into some new territory here, but also must have been a little scary to think, oh, daunting task here. I got 4 new mass specs I gotta deal with. Tell
It
us a
was.
little bit
It
about that.
was. It was. Yeah, yeah. Because I was changing gears and getting into an area I didn't have a background in, and I saw how much money the university has invested in the building, and then as far as my lab in particular with those mass spectrometers, they're not inexpensive instruments. I'm sure they were $400,000 a piece at the
Minimum.
time. Yeah, yeah. And so there was Discovery Park was kind of new, so there was a lot of excitement, and I thought, oh boy, yes, there was a lot of pressure. Um, but to me, it was fun. One thing I enjoyed was the freedom to really do the science. And one thing I haven't mentioned is working in industry. And since our the compounds we were making and approving using my methods to say yes, this meets specifications and stuff, we're going into people. They were involved in clinical trials. We were a GMP lab, so we were highly regulated. And that was another reason for kind of stepping away is uh, it's like trying to do science with one hand tied behind your back. You have to follow all the rules, and it moves progress very slowly. Um, Whereas when I came here, I was like, wow, this is more of a research facility, and nothing we're making has to be prepared at a GMP type facility or under those kinds of regulations. So it gave me a tremendous amount of freedom to do what I want. So now you can explore things in more depth.
So
So that was like being released from a straitjacket, really. And so that was part of my enthusiasm in the beginning. And it was also very fun to work on so many different projects. Where at Lilly, as I said, I would, and everybody would in my group would be assigned to one drug, and maybe you'd be some people would be on it for 4 or 5 years to complete the process. And I think it's a little dull being on the same thing. Whereas here at Purdue, at the Bentley Bioscience Center, we're a campus resource. So people come to us who need to use our skills and our technology. So I quickly found myself reading journals in veterinary medicine or plant science, as you mentioned, which are things I had no background in. And if it wasn't for the opportunity to come here, I never would have learned about. But I think one of the exciting things about being here at Purdue is just working with so many, again, people from different backgrounds, but in different areas.
yeah,
So
yeah,
entomology, I mean, you name it.
sure.
So many different things that they're working on, and I get to contribute to that. So
Yeah.
that's what's fun.
Well,
Um,
and one of the reasons why I wanted to interview you is because I wanted people to understand what it is that as a when you say, we're a research facility, people come to us and you get to do all this work with all these other professors. I wonder if you could just explain a little bit, what is a core facility? How new is this for an academic institution to have a facility where you can either send samples and have them be analyzed for you, or you can send a student to get trained. I mean, there's so many ways that people work with you. And so
mhm.
it's not only you have to be on top of the science, but you also need to be on top of the equipment and like charging and like all of this stuff that involves administration. And I mean, can you explain a little bit the life of a scientist
Yeah.
and a director in a core facility? I think people would be totally amused and in awe of what they would hear from you.
Yeah. So you asked, like, what is the core facility? So we're here to support the campus. There'd be a lot of people who realize, oh, I need to do a chemical analysis. So everything we're doing in the Bentley Bioscience Center is biological in nature. And somebody in their research might realize, oh, it would be very beneficial to, say, quantitate this certain metabolite in a plant or an insect or whatever it happens to be. But to do that, they're not exactly sure how best to get that accomplished. And so we're here as a resource. Somebody can come over here and say, here's my problem. Can you help? And we can say, yeah, we can do that. We have the knowledge base, because we're going to have to do an extraction, and we can help you think about what's the best solvent to use to target that particular compound you're interested in. So we can help you with the extraction, and then we have the instrumentation here to quantitate, and it's very sensitive, and then we can help with data analysis. So the idea of really the Discovery Park district as a whole, but Bentley, was to be an extension of a research lab. So a professor can get out of their comfort level, because they're all experts in their field. But often they need to deviate a little bit out of their comfort zones. And yes, even professors can get out of their comfort zones. So they can come over here and talk to experts. And so I can help when it comes to quantitating metabolites. There's another core facility in Bentley that looks at proteins. There's flow cytometry. There's light microscopy. There's lots of different areas that we offer. But the idea is we offer high-end instrumentation that an individual professor is probably not going to want to put a line item in on one of their grants, because they're very expensive. And it's not just a matter of buying it, it's maintaining it. I think, as you mentioned, the maintenance in my area of mass spectrometry, it's very analogous to when you if somebody was to buy an expensive sports car. You think, boy, I'm spending way more money than the average car, and I'm getting something really high performance, really cool. And then once you do that, you realize you need to buy a regular car because your sports car is going to be in the shop most of the time, getting it repaired, and you can't rely on it as a daily driver. And that's really how these high-end mass spectrometers are. They're very expensive, but they break a lot. Again, and I mentioned that as that's a deterrent for an individual professor to get one. If it's not their specialty, if they were to get one, it's probably going to break, and then what are they going to do? It's expensive to fix. So yeah, we do a lot of wrench turning here in the lab to keep these different instruments going as they age. I mean, some of them that we're using are 15 years old, so probably beyond the manufacturer's life expectancy. Some of
Yeah.
them, the manufacturers have gone out of business, and so we have nobody to turn to if we want to keep them operational, except just kind of learning to use them ourselves. But yeah, that's the core facility, and I think Purdue was kind of on the cutting edge when they decided we really need to have a facility with multiple cores on campus to support the life sciences. And now, almost 20 years later, I see it's a lot more common within different universities to have structures, institutes like this, where there's multiple cores to support the researchers on campus. Yeah. So yeah, a lot of it is grad students coming over with a problem, and we can help. And ideally, we like it if the graduate student is interested enough to be trained to do the work themselves. If it's the type of need where they might say, gee, I got 20 samples, this really isn't my area, my thing. Can you guys just run it? Sure, that makes sense. But the more common situation is where somebody comes to us early in their grad school career and says, gee, I have this need, I'm probably going to need it my whole grad school career. Can you train me how to do it? And we're happiest to do it that way, where we can bring somebody in and give them some new skills.
Yeah.
I've definitely had people come back from job interviews and say, Bruce, I have to tell you, there was this point during my job interview where I was showing data, and somebody in the audience raised their hand and said, okay, you're showing some mass spec data. I know you're really not a mass spectrometrist by training. So you mean you submitted those samples to somebody and they gave you those results? And then they would reply, no, I actually got trained on how to operate the instrument. And so I collected this data and analyzed it. And I've had students say that they could tell that their future employer was impressed by that. And it could definitely sway things during an interview process if you say, no, that's just a skill I learned.
Yeah.
And one, it
No.
shows you're willing to go outside your comfort zone, and I think
Yeah.
any employer is going to be interested in that, because
So
that's real world.
yeah, and I hope that the grad students that are watching this take a cue of that, of, put some tools on your belt, and make sure you have them for your next journey ahead, because you don't know when they might come in handy, or when you might be able to talk to somebody who understands that field really well and be able to communicate with them in a way that no other person could, right? I
Hmm.
love that, Bruce. From everything you've said, though, like, I mean, you worry about the cost of the instrumentation,
Yeah.
which you talked about that. You worry about the maintenance, and that's administrative piece. You also worry about training the student and worrying about the science behind the project that is being done. There is so much versatility in what you bring to the table, but also it seems to be like an environment where there is so much to do that you couldn't get bored very easily, you know?
Right,
Uh,
right. Yeah, yeah. People are challenging us all the time. Uh, yeah,
I
yeah,
hope
yeah.
in a good way, though.
Yeah, yeah, it is. It is. Yeah, yeah. With the things they want to look at, some things that somebody will say can. Well, I know you were looking at amino acids in the lab, and
Yeah,
so we of course people are coming to us frequently and saying, can you do an amino acid analysis? And so that's one of those on the shelf methods that we can say, oh sure, we have ways to do it. They've evolved and changed since the instruments we were using when we were working
yes,
together,
right,
when you were a graduate student. So yeah, I mean, we change things when we find better ways. We
sure,
probably just something as common as an amino acid analysis. We're probably on our fourth technique just because
yeah.
of improvements in sensitivity and minimizing sample preparation and
Yeah.
time and those things. So
And
um,
I remember, when you were training me, how much patience you had with explaining, and we had a good time in the lab. I remember enjoying my time so much when I used to come, and we used to just, talk, and you
mhm.
used to tell me a lot about the instrumentation and so on. And I learned so much from your training, not only what you were saying, but how your approach was always very, very calm and very, very much in this patient style. Did you gravitate to the training and the teaching, as you were thinking, oh gosh, I really would like to go back into academia? Is that something that you thought, oh, this is going to be fun again to do, because I might have not had that in industry?
Yeah, yeah, yeah. I mean, in industry, there was certainly turnover of bachelor level scientists, more so than at the PhD level. So we'd bring people in the lab. So I was training uh, individuals on how to run the instruments, and I did really enjoy that. And again, going back to the GMP aspect, it was very regulated. So I was writing a lot of documents. Lot of it had to be step by step. Here's what you do. Here's very cookbook.
Yeah,
Uh, uh, and so I did enjoy doing that, and I was thinking about that was a nice opportunity. Coming back to academia, where I can train younger people, I think most people find that quite rewarding. When you take somebody who's a little uncomfortable in the beginning, and then uh, they ended up being very independent users. One of the most rewarding things for me is when I go to somebody's defense. If I've worked with a student for a long time, I say, make sure you tell me when your defense is, because I like sitting in the back and hearing, and hopefully they're going to show some data that they acquired when we were working together, and just hearing them speak so comfortably about it. When I remember the days when they first walked in and they were sheepish
yeah,
and were so nervous. They understood how expensive the instrument was, and boy, I really don't want to do anything to break this. And I know other
right.
people are using it. It's shared equipment. So if I break it, other people down the road, the person who has it tomorrow can't use it. Um, but yeah, the training is nice, and so I kind of got my wish. I could train people and have be working on a lot of scientific projects. And although I do work on grants, I'm not really the lead PI. I don't have that kind of pressure, which, as I said in the beginning, was something that kind of shied me away from coming to academia in the first place. It's more fun to contribute to somebody's grant than to have the pressure of being the lead PI on
Definitely,
it.
definitely. And I know you do thrive because we've interacted together in being the PI when we write grants to obtain or acquire new equipment that you can
Hmm.
run in your facility. And I think, your leadership there, it shines through. So just so that people do understand, you do get to write some grants, you do get to participate in publications, you do
Oh
get to
yes.
participate in science from all around the university, all different fields. As you said, there's entomology, there's vet school, there's food science and nutrition, microbiology, biochemistry, biology. My gosh, you do everything, and in terms of you touching all of these life sciences at the university, pharmaceuticals and so on, right? It's incredible the breadth of science that you touch on. Do you feel like because it's really a service provider type of mentality that you
Hmm.
have to say, well, there is no my science that I'm going to be working on. It's everybody else's science that I'm going to help them with. How do you feel about that? Are you okay? Obviously, for so many years that you've been doing this successfully, and the care that you take in training and the students, this piece too must be something that keeps you excited, though, isn't it? I mean, working in all
Yeah,
the different types of sciences.
right, right. Yeah, yeah. I'm the type of person I can get excited working with somebody else, so collaborating with somebody else. It might be their project. It's working with the professor, but I can get excited by what they're doing. And you mentioned, my science. I feel I am doing some things that are just to interest me, in the sense that if we realize, oh, there's a new technique, like I mentioned earlier, we're kind of on probably the fourth generation of the way we do amino acids.
Yeah,
If something becomes popular, and I said that there's a better mousetrap, let's apply it and see if in our hands we really think it's better. So that's kind
yeah,
of an example of my science. We do have the opportunity to do our own things. Maybe there's a new derivatization approach that's different from how we've always done it before, and it might have
yeah,
some advantages. So let's explore and see if we can think that we should switch to that. That's kind of an example of us doing our science, but with the goal of how is it improving the core. It has to be a technology we can apply to researchers on campus. So that's the purpose. Um,
yeah,
but we do have the freedom to explore new ways of doing things, and that's where we have that independence of our own, and not feeling
yeah,
like we're always second fiddle to
yeah.
somebody else.
Yeah, and I think that's important though, right? Because it's the right type of character, the right type of qualities that would allow you to succeed, to be a great scientist within a core facility environment, right?
Right,
You still
right.
have to have your own wins. You still have to have your own scientific challenges and curiosities that keep you going as well.
Right, yeah, because people come to us and I don't immediately know the answer. They say, "I want to do this. Can you help me with that?" And I could think, well, I could think of 2 or 3 ways we can get that done. Maybe that's a gas chromatography experiment. Maybe that's an HPLC experiment. Maybe we could try both and compare.
Yeah,
So yeah, yeah. It's not like, oh, okay, you just have to do this, and I've done it a million times before.
right,
It's
right.
there's new technologies that we're always applying,
Yeah.
um,
No,
to keep the
that's
job interesting for us. Yeah.
yeah, because I can imagine, the giving away of the data to after the analysis, if there's something exciting there that you get to pass on the good news to the
Right.
investigator or
Right.
something, right? Oh, we finally got it. I
Right.
remember you and I having so many of those eureka moments. I would
Yeah.
come in, Bruce, we
Yeah.
got something.
Yeah. I do remember those too. Yeah.
Yeah.
Yeah. Oh yeah. And then
And
that gets me fired up. Yeah. If somebody else is
yeah,
excited and I feel like, okay, I've
I
contributed.
think I think that's what has given you such a special touch in that facility, is that you have been able to get excited about so many different thrusts. And people trust that you are part of the team and you're not
Mhm.
just there to push samples along in a machine. It's like you're interested in what they're doing, and it's been great working with you so far, Bruce, and I congratulate you on such a great success, such a great career, and it seems like you're doing what you love doing with the people that you love doing it with. I thank you also for opening up your story here on Pathfinder and telling us a little bit what the life is like in the core facility, and dealing with so many different issues. But it's been great. So thank you so much.
Thank you, Tommy. Happy to help. It was fun.
Absolutely. Take care. Have a great afternoon, and we'll speak to you soon.
Thank you. Goodbye.
Okay. Bye bye.